This is the entrance to one of the most tightly guarded and dangerous places on Earth. Here, the final confrontation is taking place in a battle that has lasted centuries, a battle that has claimed as many as 100 million human lives. Now, it is the enemy's turn to die. Locked away here at the Centers for Disease Control in Atlanta, Georgia, and at a similar facility in Moscow, are the only surviving smallpox viruses on Earth. Soon, these last smallpox viruses could be destroyed, our first deliberate extermination of another species. But even as smallpox eradication symbolizes our victory over many of the great killers of the past, other diseases, including the world's newest plague, AIDS, continue to defeat us. Every minute of every day, we're all in a battle for survival. Our enemies are countless invisible microbes that seek food and shelter within our bodies. If they succeed, they can make us ill or even kill us. We only survive this unceasing attack because we're protected by a remarkable defense. At its heart is an early warning system that can spot and quickly identify an intruder. Then, an armed response is mounted that's custom designed to counterattack the invader. This defense, our immune system, has evolved through millions of years of pitched battles with our enemies. Unfortunately, our enemies have evolved too. They're constantly probing our defenses, seeking its weak points. A major victory for the microbes came at the end of the First World War. In October 1918, as the Allies were beginning their final offensive, a new enemy struck that was deadlier by far than even trench warfare. In a few months, it killed twice as many as died in 4 years of people fighting against each other. The new enemy was a strain of influenza. Before it ran its course, it killed some 20 million people around the world. Today, every winter, the flu returns. Every winter, there's a chance the flu virus will unleash another major pandemic. Even in normal years, the flu virus infects hundreds of millions of people and still kills. Modern medicine's major weapon against the flu is made inside tens of millions of hen's eggs. Grown within each egg are inactive versions of the flu virus, from which the current flu vaccine will be manufactured. And every year, it's a race against time to produce 30 million doses.
We have a crisis atmosphere that develops for a period of about 6 months. We can make no mistakes. We must meet the urgency of the U.S. population needs and those in Europe. And our people are living and breathing flu from morning till night, well into the early spring.
Despite these annual efforts, the flu vaccine will not reliably protect us from future flu epidemics. The smallpox vaccine has. A prolonged worldwide campaign has eradicated this deadly disease. Smallpox will never strike again. Why is the smallpox vaccine so much more effective than the flu vaccine? When a flu or smallpox virus invades the body, it is met by an army of white blood cells. One whole regiment of cells is armed with feelers called antibodies. Each cell has its own unique antibody, with which it investigates the intruder. Cell after cell tries for a fit. Eventually, one comes along that's a perfect match, and it's triggered to multiply into billions of cells. These pour out the appropriate antibodies, which bind to the viruses and lead to their destruction. The system is marvelously versatile. No matter what the invader, eventually an antibody cell will come along that recognizes it. But there's a price for this versatility. It takes time for the right cell to turn up and to multiply itself. And during that time, the flu or smallpox virus can be doing its work. In some cases, the immune system wins the race and the victim recovers. Now there's protection from a permanent platoon of cells which already possess the right antibody. They're ready to pounce. It's this immunity to reinfection that is created by all vaccines. The smallpox vaccine contained a harmless virus that looked like smallpox to the body's cells and prepared them for the real thing. By 1979, smallpox had been reduced from a deadly plague to a laboratory curiosity. Smallpox killed by invading and overwhelming our bodies before the defense could be organized. The vaccine we used to beat it was essentially a tip-off that an attack could be expected, so the defenses were put on alert. While smallpox was swift and brutal, once our defenses were ready for it, it had no strategic options left. But the flu virus is more versatile, and so is still a potentially deadly threat. Coming into Atlanta, Georgia, is a little box that contains a new strain of flu virus from China. It's on its way to the Centers for Disease Control and is the latest candidate for causing a major epidemic. Unlike smallpox, the flu virus is constantly changing, so our immune system is always being caught off guard. The vaccine that worked against last year's viruses may not work against this year's. And it's up to Nancy Cox, head of the influenza branch, to recommend which virus strains to include in next year's vaccine. And there's only room for 3.
Helen, here's that fax I just got from Dr. Guo. And they've got influenza outbreaks in Beijing occurring in some primary and middle schools, and they've got influenza B. The vaccine manufacturers actually have to have all 3 strains chosen and to them so that they can begin producing the vaccine by early spring. And we have to anticipate what's going to happen the following winter. This is really rather difficult, and it puts us under a lot of pressure doing as many tests on as many viruses as we can in a relatively short period of time.
Sure, this is a test that we did.
Every year, Cox and her colleagues have just a few weeks in the late autumn and early winter to predict what the flu will do. There are 2 basic families of flu virus, type A and type B. And this year, it's type B that looks most threatening.
Illinois, Taiwan, Brazil.
We're seeing a lot of influenza B viruses circulating at the present time. And this is causing us some concern because we're expecting, on the basis of this, that we might have an epidemic of influenza B this coming season.
The virus just flown in from Guangdong in China might be different from the B strain that was used in last year's flu vaccine, called B Panama. To find out, antibodies that work against Panama will be tested against the Guangdong virus. This will be the first clue she needs to decide whether the existing vaccine could work against Guangdong.
Hi, Henry.
Hi, Nancy.
Well, what happened?
Well, take a look for yourself. I've run the new Guangdong virus against the Panama antibodies, and it's not reacting at all well.
The number of red dots shows how well the vaccine would work. The new Guangdong virus is the second row from the bottom.
Okay, well that indicates that this Guangdong virus is a new variant, and we'll have to keep an eye out for that one.
Right.
But exactly how has the virus changed so that it could sneak right past the immune defenses put in place by last year's vaccine?
Oh, this is nice. I can see the spikes pretty well here. Using an electron microscope, we can begin to see the structure of the virus, and we can actually see the spike, the surface spike.
These spikes are what human antibodies latch onto. And here is the secret of why the flu virus is constantly changing. Inside the virus are just 8 genes. The genes of most living things are DNA, where the genetic code is protected within the 2 intertwining strands. Only rarely do mutations change this code. But the flu virus carries its genes on a single-stranded relative of DNA called RNA, which mutates much more easily and often. Because its genes keep changing, so do its spikes. It's almost as if the immune system has never met the flu virus before, with predictable results.
I started working on influenza when I was a graduate student. And I think that the thing that has kept my fascination through all these years is that although we know a great deal about it, we can't predict what's going to happen next with any great certainty. It's like a puzzle.
Nancy Cox keeps frozen samples of all puzzles from the past, all the different guises the flu virus has gone under for the last 50 years. Some caused devastating epidemics. In most cases, the spike of the virus changes its shape, and this fools our immune defenses. The computer reconstruction shows how a spike is built from a complex arrangement of protein chains. Even a tiny change can be enough to make it unrecognizable to antibodies. The colored regions on this view of the tip of a spike show all the places it has changed over the last 15 years. With time running out to decide on next year's vaccine, the Guangdong virus is looking different enough from last year's Panama virus to make Nancy Cox's decision difficult.
This paragraph here. That's really all.
Should she switch to Guangdong?
Well, I think because the deadline is so near, we better get that back to Lee.
Her answer will depend on whether it looks as if the new strain is spreading from its Chinese origins. And it's already turned up in Taiwan, Cambodia, Australia, Chile, Argentina, and Brazil.
The fact that the B. Guangdong is spreading makes it an excellent candidate for next year's vaccine. But on the other hand, we have other viruses which are also a little different from the Panama vaccine strain. And we have to watch them and see if they're spreading as well. And we really work right down to the last moment, watching a complex picture in order to see which virus we believe is the most likely one to cause the next year's epidemic and get that in the vaccine in time to stop it.
In February 1993, Cox made her decision. On it may hang thousands of lives. She decided to stick with last year's Panama strain, gambling that while Guangdong is on its way, Panama still poses the greater threat. Only time will tell if she guessed right. The flu virus of 1918 that killed 20 million in a worldwide pandemic wasn't just a subtle variation on previous viruses, but one that had altered beyond recognition. Another such drastic shift occurred in 1957 when Asian flu swept the world. 11 years later, it seemed that the flu virus had completely reinvented itself again when Hong Kong flu struck. Dr. Robert Webster.
It was in 1968 with the Hong Kong pandemic, the worldwide epidemic, that the scientific community realized what was happening. Until then, we thought that the virus just drifted along, changed slowly, and at some time it accumulated a lot more changes.
But the changes in the 1968 virus looked much greater than the subtle annual changes the virus usually undergoes. The spikes looked almost alien, as if they came from another species.
There'd been reports of influenza in horses in some parts of the world, some serious diseases in chickens, some in pigs. So we set out on a worldwide hunt. What a great excuse to travel the world and find viruses.
What Webster was looking for was a virus that could be the source of those strange spikes on the 1968 Hong Kong flu virus. And in Canada, he found it, a virus that infects ducks and that carries in its genes the instructions for the Hong Kong spikes.
It's very interesting in wild ducks because the viruses cause no disease and they spread from duck to duck through the water. But you can't catch influenza virus from a duck by eating it, by handling it. So how do they transmit to humans? And do they transmit to humans? Indeed they do. The Asian pandemic had 2 of its 8 genes from wild ducks. The Hong Kong had 3 of its 8 genes from ducks. So we're very sure that the duck is involved in this picture. But studies show that the influenza virus in ducks doesn't multiply in humans. So how do they get together? We realize now that the pandemics, these worldwide epidemics, come out of China, Southeast Asia. All of the big pandemics have names associated with that area. So here we are, we have influenza viruses in ducks in Canada and pandemics coming out of China. How do we put these 2 pieces of information together?
The answer hinged on the traditional living arrangements in rural China. Both ducks and pigs share close quarters with farming families, all crowded on small areas of land. The 3 species are constantly exposed to each other's germs, and pigs and humans can even catch each other's diseases. The pig turned out to be the virus melting pot Webster was searching for. It's able to harbor both the human and the duck flu viruses. The genes of the 2 different viruses infecting the same animal at the same time can get mixed up. So a human flu virus now can contain a duck flu virus gene and a pig virus spike. In an instant, a totally new flu virus has been born.
And the immune system doesn't have time to respond to that. The vaccine manufacturers don't have time to make a new vaccine. And so consequently, the virus spreads in people, in nations and around the world, and causes this pandemic.
It's the sudden emergence of such a pandemic strain of the flu virus that is Nancy Cox's greatest nightmare.
In our surveillance, we're looking not only for the new variants as they come along that are put into each year's vaccine, but we're also looking for a new pandemic strain that might emerge in humans and begin to spread. And we want to find that new pandemic strain in time so that we could actually make some vaccine and make plans to protect at least part of our population.
Unless the way people, ducks, and pigs live together in China changes, another flu pandemic is inevitable. The only question is when. The best we can do is to stay one step ahead. Unlike smallpox, the flu virus has far too many forms for it to be eradicated by a vaccine. Bigger parasites cannot change their genes as fast as the flu virus, but one of them, the schistosome worm, beats human defenses by disguising itself and pretending to be human. 400 years ago, it came to this Brazilian port with slaves from Africa. Today, the worm that tries to be human devastates the lives of 8 to 10 million Brazilians and 200 million people worldwide.
Important is to see, to find out if
A Brazilian-American team is now attempting to develop a vaccine against the disease. Don Harn has worked on the idea for years, together with Evan Secor and Mittermeier Rice. The disease flourishes inland, where the heat is fierce and the water precious.
And right along all those houses are where the infection's the worst.
Oh, well, and you can see that they don't
back right up onto the
The town of Itacara is so dry that the stream running through it was dammed to create a lagoon. The lagoon is the town's most valued asset, yet it is also what brings schistosomiasis to its people. 9-year-old Rogerio's story shows why a vaccine is badly needed.
Nós foi caçar passarinho e daí os meninos assim. Ah, tô com calor, então bora ali no rio tomar banho, daí eles assim. Então, bora lá, daí eles assim, não, então eles assim, então bora lá e acabar. Ele foi, com a brincadeira, tirou minha roupa e jogou no tempo. Acabou, fiquei continuando a tomar banho dentro, acabar. Nós caçava de novo passarinho. Daí nós ia tomar banho no rio. Foi dia nós pegou cistosa.
When Rogerio was first diagnosed, his liver contained more than 1 million eggs from the schistosome worm.
Now we look to see if his liver.
Drugs have taken care of the eggs for now, but repeated reinfections will further debilitate and could eventually kill him.
Okay.
Reinfection is likely because of the way the villagers live. They get infected from the precious water in the lagoon. The challenge is to find some point in the parasite's life cycle which the scientists can attack.
everywhere. So they're going to be in the water.
A snail, as well as humans, is host to the parasite. This freshwater snail thrives on human waste. Living within the snail and stimulated by sunlight to swim free are thousands of larvae of the schistosome parasite. These larvae, less than 3 millimeters long, swim through the water until they find exposed human skin. Once in the body, the parasites travel through the bloodstream to settle in the veins that lead off the intestines. There they grow into 2.5 centimeter long adult worms. The larger female and the smaller male lie together, feeding off the blood in a permanent act of mating. During their lifetime, which can be as long as 20 years, they produce billions of eggs.
You've got this large organism that's a foreign body inside of yours. And so basically your immune system should be attacking it and killing it, but it doesn't. the adult worms live right in your blood system where the immune system, all the immune system factors are right there to kill it. Yet the parasites may live from 3, 5, it's been reported as long as 20 years. Why is this? Why is it that a foreign body can live in an adult that amounts a perfectly normal immune response to anything else?
The answer seems to be that written in the worm's genes is a cunning way of making itself invisible to the body's immune system. The worm bears on its surface characteristic spikes like all foreign invaders, which should label it as not belonging. It should be these spikes that are recognized by the cells of the immune system. Some of these cells coordinate mass attacks to destroy the invaders. The worm's trick is to hide its telltale spikes behind molecules it steals from its host, so that the host cells see the worm as human and ignore it. So the worm's eggs continue to cause the hugely distended abdomens of its victims by inflaming their livers. The eggs themselves find their way back via human waste to the water supply and hatch into yet another stage of the schistosome's life cycle. These larvae search out snails, and the cycle of infection is complete. In Itakara, the route that completes the infection cycle is only too obvious and suggests an equally obvious way to control the disease.
If we improve the education and if we improve the sanitation here, give latrines and teach them that they should use the latrine, I think that the transmission will decrease a lot.
As the research team explores the town, they discover that so much of Itakara's life centers on the lagoon that education and sanitation won't themselves be enough to break the cycle of infection. This reminds Don Harn why he began his vaccine research. 10 years ago, he visited a village where the people had already been educated about the disease.
When I asked them about, do they know about schistosomiasis? Sure, we know all about it. And I looked in the water and geez, there were these snails all over the place. And I said, do you know about snails? And one woman completely bent down and picked one up and held it in my face and said, absolutely, here it is. And that point convinced me that something other than educational means were needed to eradicate schistosomiasis. And it was really a gelling point to say, no, I didn't make the wrong career decision in looking for a vaccine for schistosomiasis, that indeed it was going to take a long time, but that a vaccine was clearly a way to go.
Don Harn plans to beat the schistosome by aiming a vaccine not at the adult worm, but at the larva as it enters the body and before it has a chance to take on a human disguise.
Infected at different levels.
The strategy will only work if the villagers' immune cells are able to recognize the larva as foreign. To find out, the research team collects blood samples from some 300 townspeople. The crucial confrontation between the immune cells from